excellent discovery.
Hi, I'm Wayne Curtis. I've been here for now over 25 years. The research in our lab has started originally in the plant biotech. We worked a lot with companies like Monsanto and Weyerhaeuser, doing what I refer to as applied plant biotech work. That is expressing proteins in plants, but also propagating plants. We did that for Weyerhaeuser. Also producing defense chemicals and things like pesticides. That work then also involved working on medicinal plants and trying to produce drugs that were naturally produced in plants. And the work then from there morphed over a little into the world of algae. Plants and algae obviously are somewhat related. And that work on algae and photosynthetic systems led us to work on more of the fuels side. We worked with an algae eventually that was a hydrocarbon producing algae, and then with collaborators we obtained the biosynthetic pathway. And it's a characteristic of our lab is we tend to work a lot with with scientists who are specialists in that. So Joe Chappelle was the one who had done that work. That led to more biofuels work of genetically engineering the biofuel synthesis pathways into unusual organisms, not just like E. coli, but organisms that could consume hydrogen and oxygen as autotrophic work. The algae work also led us to work with photosynthetic bacteria. And the photosynthetic bacteria then, where the work was primarily initially focused on reactors, we also were interested in then using those reactors for growing algae or bacteria to produce membrane proteins, because photosynthetic bacteria do that. And so our research then and has continued to work on expressing functional membrane proteins in bacteria. And some of the work in the fuels then is also then encompassed working with cellulosic biofuels, because if you're producing biomass, either plants or algae, even if you have 30% oil, you're still going to have 70% that is actually the biomass. And so we've worked on cellulosic biofuels. And our approach there has been to use more than one organism, one that's been engineered for the consumption of the biomass, and the other that's producing the fuel, including the hydrocarbon biosynthesis pathways. And so that, if you take a look at the big scheme of things, we work at the molecular level, our plant propagation work, which now includes manipulating plants with transcription factors. To the bioreactor scale, I happen to be standing next to a large fermenter, and in chemical engineering, the bioreactor is the house that you put the organism in, and you can control the environment. And so that environmental control now extends to genetic control. So you can turn genes on either by controlling the physical environment or the actual genetic environment. So that ranges from plants, where we're turning on the process of making embryos, to the bacterial world, where we're trying to understand how the bacteria work together and stick to the cellulose to break down the actual product itself. That's it.
Hi everyone, this is Dr. Mustafa Arbaka. I recently completed my PhD studies in CurtisLab, working on the development of Rhodobacter sporoides as a functional membrane protein expression platform. So membrane proteins are extremely important for all living organisms, as they carry out crucial physiological roles, including energy generation, signal transduction, and transport of solutes. And they also comprise more than 60% of the drug targets in the pharmaceutical industry today. However, the membrane protein studies are hampered by their low availability in the natural sources. Therefore, we need to overexpress membrane proteins to get more information about them. Traditional expression hosts, including E. coli, has limited membrane area for expression of membrane proteins. Therefore, we've been looking for alternative expression hosts, and we decided to employ Rhodobacter sphaeroides for membrane protein expression. And this was because Rhodobacter has 1 to 2 orders of higher membrane surface area available for membrane protein expression. This is because it's a photosynthetic bacterium and the photosynthetic apparatus of Rhodobacter sphaeroides are naturally accommodated on those membranes. And using this system, here I can show you the ring there growing Rhodobacter using this infrared LEDs. We were able to express membrane proteins from different organisms and classes, including human weather channel proteins to multi-segment enzyme complexes. And this pretty much sums up my research.
Hi, I'm Dr. Langstaff, and I work on the membrane protein expression research group, which is with Mustafa. And what we do is we grow Rhodobacter sphaeroides in a photoheterotrophic system to yield membrane proteins. And some of the membrane proteins we work with are aquaporin. We work with a Rhodobacter aquaporin and a human aquaporin. And I guess what I do is try to work on the platform to better it, and I do 2 things. The first is I made a pH control system for our photobioreactor setup, and the problem with the old setup is that undefined medium would become too basic and it would stop growth at a really low biomass yield. So I created a pH control system using a pH controller and succinate at a pretty low molarity of around like 0.25, and this controls the pH, increases the biomass production, which in turn gives you a higher volumetric productivity on the protein that we're trying to grow, which is usually things like Rhodobacter aquaporin and the Rhodobacter aquaporin with the membrane or with the M-banana fusion protein on it. And then we usually test the functionality. And unlike most people in CurtisLab, I do downstream stuff as well as the upstream stuff. So I purify the protein and work on purifying techniques, and then work on helping Ting Wei, who's in Kumar Lab, with functionality. And then the other project recently that we just started is working on a new plasmid. And the other project I'm working on with the Rhodobacter platform is working on the plasmid to get rid of a couple things and increase the productivity. So we want to get rid of the tetracycline resistance gene, and with the plasmid we just got called PRKD418, it has a trimethoprim resistance gene. And the reason we wanted to go away from the tetracycline is because when UV light hits it, it photooxidizes and releases, well, it turns into a toxic substance that will kill the bacteria. Yeah. And also, the emission spectrum of tetracycline interferes with the fusion fluorescent tag proteins, like the M-banana or EvoGlow, and I think there's one more, but I don't remember. And so this new plasmid has the trimethoprim resistance gene, which is an antibiotic that doesn't interfere with the emission spectrum of the fluorescent tag proteins and isn't affected by light, so we aren't restricted in our photobioreactor system. And I think that's about it.
I'm a graduate student in Dr. Curtis group. Today we are going to show you what the Algae group is currently doing, and our many go through 3 projects. First is about the pH control. The Secondly is about the algae film reactor, and thirdly is about some exenic culture work on Botrycoccus browni. To the shaker here, you can see experimental setup for our pH control experiment. So what we are currently doing here is we are setting up a pH state so that we can measure how much the algae is producing the hydroxyl or proton during their growth. And with that knowledge being measured of being controlled, we could actually predict how much acid or base or different fertilizer we can feed into the system as algae is growing up. This is kind of fundamental work, and for the starting to figure out the starting metric ratio of algae growth. And after we measure those data, we actually prove that algae could be actually grows, and by slowly feeding them to nitric acid, and this even makes the pH go higher. So this becomes the foundation of our future pH control algorithm and the high density culture stack. The second thing we are going to go through is about the film reactor, developed in our lab for several years. And right into here, you can see a kind of bag reactor. And the general idea is that we are going to maintain the algae culture in a small container, and this container, the algae suspension inside the container will be pumped and then go through and enter this reactor from the top, and then they trickle down through the film and all the way down to the bottom, and then go back and recycle to the suspension container. So everything, the algae culture will be keep recycling and moving through this reactor. And meanwhile, the gas will go through to the other side, and then the gas and the liquid algae culture will finish their transfer and exchange to almost mesh structure. If you go through, if you come closer to the film, you will see that those mesh structure looks quite complicated, and the purpose of them is to generate a very high gas to liquid mass transfer coefficients. So with this kind of reactor, we actually optimize all the gas exchanger and the light penetration condition, and we actually reach about 20 grams dry weight per we actually reached about 20 grams dry weight of algae per liter, and which is so far to our knowledge the highest value of our liters. Here is a culture system we built to maintain the existing culture of Botrycoccus browni, together with the root culture. So the purpose is let the algae to produce oxygen, and in return the roots will consume the oxygen and generate the carbon dioxide to provide the culture of the algae. So the Botrycoccus, why we do this is because that Botrycoccus is actually a very promising algae that many people want to use them, but the bad thing is that this culture grows very slowly. So right now a lot of people is trying to do transgenic work to improve their property. However, since nobody actually ever gets exenic culture, so our lab then spent several years to figure out how to maintain the exenic culture of Botrycoccus. After we obtained the first exenic culture in the world, we actually found that for this strain, if we use traditional way to maintain them, they become non-exenic anyway. So as a result, we invented this way to keep them exenic, to keep the exenic culture. So and the next step of this project is that we are really going to do some transgenic work to try to improve the property of this algae, so to see what we can do in the future of algae downfield.
Hi, my name is Naimul Khan, a member of the CurtisLab. My project is on biofuels, basically. It's renewable biofuels. It's one of the projects funded by ARPA-E, Electrofuels program. So the idea is that the energy from the sun to be captured in some kind of liquid fuel molecule that we can use for transportation fuels. For example, the electricity is generated in solar photovoltaics can be used to split water, generating hydrogen and oxygen, and the hydrogen, oxygen, and CO2 can be fed to a bacteria that is genetically engineered to produce a fuel molecule. In our case, we chose a hydrocarbon fuel molecule because of its hydrophobic characteristics. You can separate this from the water phase very easily, so avoiding the separation cost. And then this is much more similar to a petroleum crude that we use in the petroleum industry, so you can feed it directly to there and then get your gasoline product. So that was our fuel molecule. It's botrycosine. It's a C30 hydrocarbon. And we chose a bacteria called Rhodobacter capsulatus that can use hydrogen and CO2 and oxygen and then we've genetically modified that to make this fuel molecule. We have achieved significant improvements recently. We've gone to 100 milligrams per liter production of botrycosine from H2 and CO2, feeding this to this bacteria. And we're working on making this system much better.
Other stuff related to ARPA project, where I was trying to design a mega plasmid which has a size of 19 kb. It was supposed to be conjugated into the Rhodobacter capsulatus, but it didn't happen to be the case because there were problems over the amplification of the pieces which we were trying to do PCR and then construct the mega plasmid, but it didn't work currently, and it's not working. But it's basically mega plasmid construction using the Gibson assembly method. There are 5 pieces in there. So far we have amplified 3 pieces. There is some other guy known as Justin Yu who worked on the 3 pieces, and the other 2 pieces we are currently working on them, but it's not yet successful as it was supposed to be. But we recently figured out that there is some problem over the vectors which we are trying to amplify. The vectors aren't there. What we are thinking to be is there is not there. So that's the mega plasmid project that I was working on, and I also was helping Justin trying to do some genomic DNA extractions and some simple stuff, and learning from him some molecular biology skills.
My name is Gabriel. I'm an exchange student from Brazil. I came to CurtisLab during the summer semester. I have been working here for 1 month, and during this time I have been working mainly in a project related to syngas fermentation, which a bacteria called Clostridium inundale is able to produce ethanol and by synthesis of different gases, mainly CO, carbon monoxide, CO2, and hydrogen. So this bacteria changes this gas into a liquid fuel, in our case ethanol. In another project we have been working consortium fermentation through different bacteria, C5, and also in this project we have been using in this fermentation yeast and the other enzymes in this fermentation.
My name is Sergio. I'm a 4th year grad student here, and I'm in charge of the plant propagation projects. And pretty much what we're trying to do is we're trying to come up with ways to more efficiently produce elite varieties of plants. And in particular, working with cacao, we're collaborating with Dr. Gilton's lab over in plant biology. And what we're trying to do is essentially clone one plant that has a genetic trait that we want, such as disease resistance or productivity. And the idea is to take one of the cells and you submit it and you expose it to a transcription factor that will turn on pathways that eventually will lead this. Currently I'm working with a lot of transcription factors, and one of them in particular, baby bloom, has the ability to induce all these pathways. So we see that when this gene is overexpressed, now you have somatic cells go directly into creating new embryos. The second aspect of the project is to come up with a platform to produce these embryos at a very cost-efficient manner. And the idea is to generate these reactors so that the cost is the lowest possible. And the method we're using is this temporary immersion system where the tissue is slowly submerged in nutrients for a small amount of time, and then this eliminates the mass liquid boundary layer, so our oxygen flux is a lot higher, so we're able to grow these tissues where it's pretty large in diameter. And the thing that makes our reactor different than other systems such as the Brita one is that we use gravity to push the liquid back, and we use the siphon to bring it back into the media reservoirs. And this allows us to decouple gas and liquid, which by doing that, now we're able to manipulate the headspace to a very precise composition with very low flow rates. So by combining a transient transcription factor strategy and a low-cost reactor system, we're hoping to develop a process that we can very inexpensively produce lots of plants that have a desired trait.
My name's Tina Lai, and I am a master's student in the plant biology program, and I am currently working in the CurtisLab group. My project is on characterizing the effects of PLA1 protein in Theobroma cacao somatic embryogenesis tissue culture system. So, first I want to explain about Theobroma cacao, which is this plant right here. It is the chocolate tree, and this plant, basically what I want to do is I want to use this protein called PLA1, which stands for phytocyanin-like 1 arabinogalactan protein. This special protein, when you add it into plant tissue culture media, it induces embryo formation, and which improves somatic embryogenesis. Now that I know I threw a lot of terms at you that you might not understand, so I'm going to explain to you. Somatic embryogenesis basically is the process of producing plants from non-sexual organs, which are on a plant are things like leaves or flowers or stems, just basically any cells that are not part of the seed. As you can see here, there's lots of leaf cells, there's lots of flowers, and all those can be used to produce lots and lots of baby embryos, which will produce lots and lots of baby plants. And this special method of producing plants creates clones of plants, where when you find a plant, like a tree that is disease resistant, you want to keep that strong disease resistant trait. And actually, in the chocolate tree system, many chocolate tree farms are being devastated by plant diseases. Over 50% of chocolate tree crops are destroyed by diseases every year. And a lot of tropical countries rely on the chocolate tree as their main economic resource. So if we can improve this cloning system to create stronger chocolate trees, then we can really help these farmers that rely on this for their livelihood every day. So an example of how the normal propagation system by seed is, you can see here, these are chocolate trees that are produced from seed, and as you can see, like, many of them have been planted, but only some of them are growing up. However, with the somatic embryogenesis system, we can get lots and lots and lots and lots of embryos from just a tiny bit of a leaf, or like a tiny bit of a flower petal, and you get tons and tons of embryos. So what's special about how I want to improve the system with phytocyanin-like 1, or PLA1 protein, is first they discovered PLA1 protein in the cotton somatic embryogenesis system, and in the cotton system they found that when you added this protein into the media, like the callus were able to produce more embryos, a lot more embryos than the process without the protein. And that's what I want to do in the chocolate tree system. My project is split into 3 sections. The first section is involved in molecular biology, where I find the orpholog, which is the identical counterpart of the PLA1 protein in the chocolate tree system. I identify the PLA1 genes in the chocolate tree system, and then I clone them into these protein expression vectors called PET14B. And after the cloning is done, there is a protein expression aspect of my project where I produce the protein using E. coli.